Next we'll see how the enzymes act or nature of enzyme action. Now the enzymes bind to the substrate at the active site, okay, to form an enzyme plus substrate, okay. Next they become one compound and it is called as an enzyme substrate complex. First it is E plus S, just a transient combination, right? Next it becomes one with each other. That is, a chemical bond is broken and all the reaction is happening actually. Then it is what is formed? Enzyme substrate complex, right? Next, after this chemical reaction, a new substance is formed, which is the product. It is called as enzyme product complex. Then lastly, the product is released as well as the enzyme is separated, E plus P. So first is E plus S, enzyme plus substrate, then enzyme substrate complex, then enzyme product complex, then lastly we get enzyme plus product. Okay. Now see this, I already told you about the lock and key mechanism. See here, the enzyme which is called, this is the active site, like this one, okay, active site, and to which the substrate gets attached. See here, this one, this place, okay. So lastly you get an enzyme plus product. Clear? Children, we know that the enzymes are used to speed up a chemical reaction. So how do they speed up the chemical reaction? Okay. Now for a chemical reaction to happen, there is a certain kind of energy is required, which is called as the activation energy. For both catabolic and anabolic reactions, there is a energy level or energy state which is called the transition state energy, which is at a higher energy level. Okay, in an exothermic or an endothermic reaction, the substrate has to go through a much higher energy state called the transition state energy. Therefore, activation energy is the difference between the average energy and the transition state energy. Activation energy is the difference between the average energy of the substrate and the transition energy. Okay. Now, if the product P is at a lower energy level than the substrate, the reaction is an exothermic reaction. Exothermic means which releases energy, you know that. Okay. And in a biochemical reaction, enzymes, actually what do the enzymes do? The enzymes lower the activation energy. It will help to reduce or lower the activation energy and thereby speeding up the reactions. Okay. Now you should know what are the different factors that affect the rate of a reaction or enzyme activity, which is one of the factors is temperature and pH. Another is the concentration of the substrate. And the third one is the presence of an inhibitor. Okay. Now first one we'll see about the temperature and pH. What is our normal temperature? Our normal body temperature, 37 degree, right? So that is the optimum temperature for all the different types of enzyme acting in our body. So every reaction has got an optimum temperature and pH also for its action, right? What will happen if the temperature is increased during fever, or fever and all, our temperature rises? What will happen to the action of enzymes now? What are enzymes now? Enzymes are, they are proteins. And what will happen to proteins at high temperatures? They get denatured. And what about low temperatures? At low temperature, the enzymes, the proteins, get inactive. They become inactive. Clear? The same thing happens in our body also. At high temperature, they get denatured, cannot act, and at low temperature, they are inactive. So enzyme shows higher activity at optimum temperature and pH. Activity declines below and above optimum value. At low temperatures, the enzyme temporarily becomes inactive. At high temperatures, enzyme gets destroyed because proteins are denatured by heat. Now you know that you have studied in the bacteria which live in extreme type of climatic conditions. What do you call them? Yes, they are the thermoacidophiles. But they also have biochemical reactions in their body, right? And they in their body they have enzymes which can act at high temperatures. Okay, right. So inorganic catalysts work at high temperatures and pressure, but enzymes get damaged at high temperature above 40 degrees Celsius. And thermophilic organisms have enzymes which are stable at high temperatures up to 80 to 90 degrees centigrade. Okay, so this is the graph which you have to draw for the activity, for the temperature and pH of activity of enzyme, right? First it increases, then it slowly decreases, both the cases temperature, because this is the optimum level. This is the optimum level. Okay, right. Now, what is a concentration of substrate? Now, when these enzymes are added to the substrate, product is formed, and as well as enzyme is again got back. Okay, if we increase the concentration of substrate, what will happen? The rate of reaction, the speed of the reaction it increases at first, and that is called as a Vmax. First it increases to a greater height, greater level, which is called as Vmax. Then later slowly it slows down. Why, children? What happens? Because there is the enzymes is getting over, has got reduced, but the substrate molecules are more. So the substrate has no enzyme molecule to react with. Okay, thereby the slows down. Okay, so you have to draw this diagram for this graph for the concentration of substrates. Clear? Which is this Vmax. This is the Vmax value. Okay, right. This gives the velocity of the reaction, and this gives the concentration of the substrate. Clear? Next is another very important and interesting thing about the presence of an inhibitor. How the enzyme activity changes in the presence of an inhibitor. Now you know about the lock and key mechanism of the enzymes, right? So in our body there are some substances which mimics the shape of the substrate. Okay, for example, there is the enzyme succinic dehydrogenase. It's an enzyme which converts, or which acts on, which should react with malonate. That's a substrate, right? But there is an inhibitor called as succinate, which is similar to malonate. Okay, so it is inhibited. The reaction is inhibited because they look same, and the enzyme cannot differentiate. Okay, now where do you find such kind of things, children? Our vaccination. What is vaccination? Is giving the antibodies, already prepared antibodies, or the killed germs are injected into our body, which will, so our body can produce antibodies prior to the disease, we getting the disease, right? So there it is useful, this inhibition, right? Okay, so this is one example. There are many examples in our body, children, of an inhibitor, presence of an inhibitor, and thereby what happens? The reaction is not happening. Clear? So this is called as competitive inhibitor. Right? And competitive inhibitors are used to control bacterial pathogens, similar to vaccination I just told, isn't it? So these pathogens are modified to you will learn it into a standard in detail. Just you should know, okay, competitive inhibitors are used to control bacterial pathogen. Now next is the nomenclature and classification of enzymes. All enzymes are proteins, and enzymes help in speeding up the reaction. Okay, but according to the type of bond it breaks or it joins and things, they are given different names, right? You should know what are these different kinds of enzymes. Most of the enzymes are hydrolases, children. Hydrolases. Right, hydrolases, for example, what do hydrolases do? They catalyze hydrolysis of ester, ether, peptide bonds. What is a peptide bond? Between two amino acids, right? Glycosidic. Remember RNA and DNA, glycosidic linkage. C and C, between C and C. CN halides. What is a halide? Chlorine, fluorine, iodine, no? That group, that is the halides, right? And PN bonds. Okay, so most of the reactions in the living system or the enzymes are hydrolases. Okay, and there are oxidoreductase or dehydrogenases. You know the reactions usually when one compound is oxidized, the other compound or the other molecule get reduced, right? Okay, you know that, right? So which is also called as dehydrogenase. Okay, transferase, which catalyze transfer of a group other than hydrogen group. So what do transferases do? They help to transfer a group other than hydrogen group. Clear? And there are lyases. Lyases means cut. Actually all enzymes are doing the same, but still lyases means catalyze removal of groups by mechanisms other than hydrolysis. Okay, simply you remember catalyze removal of groups. You should write all these things, children. Clear? And isomerases. What is isomerase? You might have, if a chemistry teacher has started, organic chemistry, you might have known what is isomers. Okay, geometric isomers and optical isomers. Isomers means they are mirror images of each other. The molecules are mirror images actually. In those molecules, they catalyze interconversion of optical, geometrical or positional isomers. Just you know that, children. Just you know. Isomerase, what do? They interconvert the optical, geometrical or positional isomers. Clear? And ligases. Lyases split, ligases attach. Okay? So catalyze the linking means joining of two compounds together. Enzymes, for example, enzyme which catalyze joining of bonds like C and O, P and O, etc. Right? So just you should know what are the types of reaction. And just these examples you write here. Clear, children?